into this topic of how vaccines are prepared, we need to know how a virus infects our body, and to be precise, how it enters our cell and hijacks it. And hence for that, we will see how the biomechanism works inside our cell. So first thing which we should know is that our body is made up of trillions of microscopic building material called as cells. And to be precise, approximately 37.2 trillion cells in our body. And each cell will have a nucleus. And if we zoom into that nucleus, there will be a ribbon-like structure called as chromosomes. And the number of chromosomes varies according to species. In case of humans, there are 23 chromosomes. 2 sets of chromosomes to be precise, one set from the mother and one set from the father. These chromosomes, if we closely watch, in these chromosomes are the genetic material called as the DNA or deoxyribonucleic acid. This deoxyribonucleic acids are nothing but the blueprint of who you are, or the tool through which the genetic information across billions of years that has been passed along generations, or it will be passed along for further billions of years. And if we see how this structure is designed, the structure has, if you see what is this deoxyribonucleic acid, is a DNA is a ladder-like structure with the 2 columns, and these 2 columns are formed by sugar phosphate backbone, a sugar phosphate backbone. So, and under this or inside this, the steps are formed using nitrogenous bases, and these nitrogenous bases come in 4 varieties called as adenine, guanine, cytosine and thymine. And this adenine, guanine, cytosine, thymine does not exist in singularity. That is, it will always be paired with the opposite nitrogenous base. For example, adenine will always be paired by thymine, and guanine will always be paired by cytosine. So cytosine again, so G, again A. So this kind of complementary base pair exists between these nitrogenous bases. And if we see, the DNA is comprised, if we assume that this DNA is comprised of this ladder-like structure which is formed using this sugar phosphate backbone, and the steps formed using adenine, guanine, cytosine, thymine. This structure runs, for example, to an extent of 3 billion base pairs. So there are approximately 3 billion steps in this ladder. And in this 3 billion step, for human beings, it is said that there are 20,000 to 25,000 genes. That is, these base pairs are divided into different genes. And what is a gene? Is that a gene is the information through which your trait gets manifested. That is, what trait you have outside, your outside appearance is because of the gene which you carry in your DNA. Example can be anything: your eye color, hair color, the way you behave, your IQ even. In most of the cases, not one gene, but for IQ, your height, everything, there are many genes which work together in order for you to have that trait. And hence each person varies from each person varies according to this gene composition. And the point which we now have to consider is how this gene which is present in that DNA gets transcribed into that particular trait. So for that, there is a process called as transcription, where, for example, if you see the nucleus which has this DNA or the chromosomes which are present in the nucleus containing the DNA. The information, for example, from one particular gene for manifestation of a particular trait gets transcribed into a messenger ribonucleic acid, that is messenger RNA. So if you see, similar to the way DNA is there is one other biomolecule called as RNA, ribonucleic acid. So DNA is deoxyribonucleic acid. So in simple terms, what you have to know is that, so the information we have about a particular trait is present in these genes, and in this gene has to be translated into that particular trait, and the mechanism that is used by our body is called as transcription, and we have a carrier who is called as the messenger RNA, and this messenger RNA takes that information into a structure called as ribosome, which is the place where proteins are built. So the information which is present in the nucleus gets translated into a particular protein. For example, the eye, similar, the eye color, hair color, hormones, how your body muscle composition is, or everything in your body is because of the protein, and hence that gets translated into your outer appearance, and a collection of all these traits and a collection of all these proteins makes a human being. So this is the basis. So basis of how a human being gets manifested into what they are in the outside world is because of the mechanism through which the genetic information, the blueprint which is present inside your cell, gets translated into the respective proteins. Now we have to see the next thing, how this mechanism which we have described right now is used by the viruses to hijack your cell. So the virus as per se biologically is not alive. The virus, what scientists say is that it does not have a specific mechanism of its own to manufacture its protein. Rather, what it does is that it invades another person's body and cell, uses that mechanism for making the protein it needs for its replication. So it's like the virus does not have any house for its own. Rather, it comes into our house and it makes use of all the things which are inside our house for the benefit of its own, not for the benefit of us. So how it does? So let us see. So in case of, for example, there are so many viruses. So in case of coronavirus, let us assume this virus has a structure where it is circular and it has a spike. So this is called as the spike, and this is the picture which we often see in TV. And what this spike does is that it gets attached to the outside, or it gets attached to the outside of the cell. So in this manner it gets attached to the cell, and once it gets attached to the cell, it is engulfed into the cell slowly, and once it is engulfed, it releases the genetic material. If you see, the genetic material has been of the virus. The genetic material of the virus has been injected into your cell, and once this genetic material gets injected into the cell, it goes into the nucleus, and in that nucleus it makes use of the same transcription procedure which we have described right now. The same mRNA or the messenger RNA, your messenger RNA, will be now used by this virus to make proteins for itself. So in that case, what it does is that it is hijacking your cell, and by hijacking, what it is making is that it is replicating itself. So one virus becomes so many other viruses, and it starts proliferating and it starts increasing in number. And that is why, for example, a person, once it gets affected by a virus, it can be transmissible. That is, that person can transmit to so many other people. And this is how virus hijacks your cell. So now what we have to do is that, so a virus, so this is where the survival of the fittest comes for in nature. Virus starts invading our cell. Now we have to protect ourselves from the virus. So what we do is that we use something called as signs. Signs which we discover are used for fighting all these viruses, and one such science is what is called as the vaccines. And this vaccine was first discovered, or it has been there for a very long time, but in regular terms it was discovered by a person called as Edward Jenner in case of smallpox. So Edward Jenner had many patients who were, so he was there and he had many patients who suffered from smallpox, and smallpox happened to be a very detrimental disease, and it was caused by a virus. At that time it was not confirmed which virus and what and all, but what he found was that in the patients who were suffering from smallpox, there were also other persons in the same area who had cow shelters, or who were very close to cows. And what he found was that these people who had been in the cow shelters, or had cows, had been affected even earlier. So they have been affected even earlier by a lighter version of smallpox, which was called as the cowpox. So this cowpox was not that detrimental. This cowpox was relatively manageable than the smallpox which was affecting in an adult time. So what he found was that this cowpox, or the pus from the cowpox, can be used on people, so that they may be infected or they may be affected in an earlier stage, and once they are getting affected, they do not get the main smallpox, is what he found. And this is nothing but the concept of vaccine. So vaccine is like a practice match. Having a practice, we do not have the complete fast bowling or the strict rules and regulation before we go into the original match. Why we do that is that to get accustomed, to know how the match is going to progress, and what and all we can do, and how we can be able to win over the person who is going to defeat us. So in the same way, in the case of virus, we have a practice match with less intense virus. Like for example, in the case of smallpox, cowpox happens to be a less intense virus, where we practice this and we get immune to that main virus. So this technique is what is the basis of vaccine, and how this has been used, for example, in fighting COVID virus, is what is the most very ingenious discoveries which have happened in the recent time for human civilization. In the case of first, we will see what is a Covishield, because Covishield is what is present in India, which occupies nearly 80 to 90% of the market, and it is done by a company called as AstraZeneca, found by Oxford University. And how this vaccine works is very simple, in the terms that what we do is that we use the same mechanism as the virus which hijacks our body. We use that mechanism for making a vaccine. So we are in turn using viruses mechanism for preventing them from infecting us. So how we do that is that we take a virus. For example, we in this specific case, we take an adenovirus. Adenovirus is a virus which affects the chimpanzee, and it looks similar to this hexagonal feature with this spike. And what we do is that we remove the information. This is the genetic information which is present in the virus, and what we usually do is that we remove this genetic information so that it becomes harmless. This is the point. Everybody gets very discouraged or scared because they are thinking that we are sending in a virus to infect our body. No, in the case of virus, we make them harmless in the initial step itself. So we have removed the genetic material. Then what we do is that we put in the spike, that is we put in the information of the spike of the coronavirus. Points we have to notice that first we have removed the genetic material responsible for multiplication of the virus from the adenovirus. Second, we are replacing that information with the spike protein of coronavirus. In that case, we are not sending the whole information of the coronavirus. Rather, we are only sending a part of the information of coronavirus, which is the spike protein, into the adenovirus. And what this adenovirus, then we inject it. Once we inject it, this adenovirus, same way as we saw in the how a virus infects our body, same way this virus gets attached to our body cell, and once they are attached to the body cell, they enter or engulfed, or they are engulfed into our body, and that genetic information gets inside our body. Once this genetic information gets inside our body, as you might be aware, our body transcribes. So we use a messenger RNA and we transcribe that information to make proteins. But in this case, you see this, in this case the virus has not entered completely, or we have not made the whole virus enter our body. Rather, we have made only the spike protein enter our body, and once that spike protein enters our body, this spike protein or the mRNA makes in the ribosome, these spike proteins are produced by the mRNAs which are sent into the ribosome, and once this spike proteins are produced in the body, we will find that our body is getting ready, or the body finds that this spike protein which is present is not the feature of our own body, and hence starts making something called as the antibody. These antibodies are like a mechanism for fighting these viruses. So a Y-shaped, so similar structured Y-shaped antibodies are formed in our body, and we get, this is the practice match we do, and we get ready. And this is why we have that initial fever and everything. So once we get ready, our body starts producing more antibodies and we get ready. And when the original coronavirus, so when the original coronavirus with the full information comes, this Y-shaped antibodies gets attached to this spike which is there. So if you see it, it has already, how it finds is that it has already found how these spike proteins are, or the information of the spike proteins have already been given through that vaccine, and hence that they get attached very easily to that spike protein, and hence they kill them. And this is what is called as the immunity, and this is why vaccines are prescribed for everyone. So this is the mechanism by which Covishield acts, but Moderna. And what we do in Pfizer Moderna is that we don't have any virus like the adenovirus which we use in Covishield. Rather, we make directly. So if you see in the transcription process, the genetic material gets transcribed using an mRNA or a messenger RNA. So what in Pfizer Moderna we do, we make an mRNA and we coat it with a lipid molecule and directly inject into the body. So in this case, we are not using any virus or nothing, no material is used. Rather, we make an information of the mRNA. What information we make is that we make the information of the spike protein. So coronaviruses, the spike protein information is fed into this mRNA directly, and that is sent into your body, and that gets into your body, and once this gets into your body and the ribosomes pick up that mRNA, so next step is the same. Next step, if you see, once that ribosome picks up it, same way the spike proteins are produced and your body gets ready for the foreign agent which has come inside your body and starts making antibody like this Y shape. And the same way, when the coronavirus enters, it finds the spike proteins structure suitable for destruction, and this is what is called as the immune system of your body acting on your own strength. So vaccines are nothing but your own body acting on the strengths of your body to kill the virus. So what we have done using science is that we have just made sure that there is some practice match before the original battle takes. The vaccine is Johnson & Johnson. So there are so many vaccines. So we have seen Covishield, Pfizer, Moderna. The next vaccine is Johnson & Johnson. And same way, in Johnson & Johnson, the only change is that here we use an adenovirus. So we use an adenovirus, but this adenovirus seems to be causing this virus is peculiar to humans. So in Covishield, if you see, we used adenovirus for injecting the genetic material or the spike protein into our body was of a chimpanzee. So here we use a human version of adenovirus. So next is the Sputnik vaccine. It is a vaccine prepared in Russia, where the same way, so the same adenovirus is used, the same adenovirus and the same mechanism is used, but what small change is that Sputnik uses 2 doses, and in the first dose one type of adenovirus is used, and in the second dose another type of adenovirus is used. So 2 types of adenoviruses are used for sending the genetic information into your body. So the last vaccine is the Covaxin manufactured by India. This is an indigenous product, Bharat Biotech, with the help of Indian Council of Medical Research and National Virology Institute have been able to find a vaccine for specifically for India. And how this vaccine works is, in simple terms, we have this coronavirus. We take this coronavirus and we remove this genetic material. So the genetic material which is present and inside the active coronavirus is killed. So this genetic material is killed, and this coronavirus is inactivated. So it becomes inactivated, and then it is made to be injected into your body as Covaxin, and once it is injected your body, you get immune system or practice before the original virus gets inside your body, and then we can have immunity for this virus. And if you see, the following vaccines which we have discussed right now have been used across this world. For example, Oxford AstraZeneca is used in nearly 177 countries. So Covishield vaccine which we are using is being used in 177 countries. Pfizer, Moderna in 103 countries, and then there is something called as Sputnik used in 45 countries. Covaxin is being used in around 6 countries. Johnson & Johnson is used in 24 countries. So if we see, the reason why we made this video is that to show that this vaccine which is present, this is the mechanism through which the vaccines operate. And if we see, the vaccines are being administered all across this world. The whole world is involved in this process for making or removing coronavirus as fast as possible to get into our activity as soon as possible, and to be able to go to cinema theaters, to be able to go to schools, colleges, to start work, to get to the next level of human civilization. Used our human science and our whole effort in this process of making this coronavirus as inactive as possible and to remove it from our lives. And I hope this video would convince you that the vaccines are safe, and if you have not gotten vaccinated, please do get vaccinated, and please protect your family members as well. And I hope we tide over this coronavirus pandemic as soon as possible. Thank you.